Good morning, good
Good
afternoon,
morning.
or good evening, according to your time zone. I welcome you to this fifth lecture of the online training series, Observing Marine Ecosystem Structure and Function, from Microbial Communities to Fisheries and Human Health. This is the virtual component of the in-person training course going to be held in India at the end of October, preceding the TREVOR Symposium. Apart from the students going to attend the in-person course, this series is open to the whole marine research community, especially encouraged our students and early career professionals. So we're very pleased to have such a large audience today. And the reminder, this is a lecture, so please take advantage of the knowledge of the speaker as well as the panelists, asking as many questions as you would like. And please post them on the Q&A box, and we'll have time at the end of the talk to get to them. So now, before introducing our speaker, we have a panel to contribute to the discussion, and it's composed of Heather Bowman, who is a professor at the University of Oxford in UK, Mariana Struco. She's a senior researcher at the National Institute of Fisheries Research in Argentina. Dr. Anas Abdulaziz is a scientist at the National Institute of Oceanography in India. Dr. Shubha Satyendranath is a senior researcher at the Plymouth Marine Laboratory in the UK. And Lilian Krug is a coordinator for POGO in Portugal. And now it's my pleasure to introduce our speaker and colleague and friend, Osvaldo Ulloa. Osvaldo is a professor of oceanography at the Universidad de Concepción and director of Chile's Millennium Institute of Oceanography. He's an internationally recognized biological oceanographer. His research has advanced our understanding of marine microbial biodiversity, environmental genomics, and ocean biogeochemical processes. In recent years, he has played a leading role in the exploration and scientific study of the Atacama Trench. And he's going to be talking about exploring microbial communities through molecular approaches. So, Osvaldo, the floor is yours.
As I said, thank you very much. Sorry with my voice, but I have a cold right now. It's winter here in Chile, it's rainy. So please keep with me. So I've been asked to give a talk, a lecture about how we can use molecular approaches to understand microbial communities in the environment. Of course, as an oceanographer, I use the ocean, but most of the things that I will say can also be used in any aquatic system or in soils or basically in any environment. And one of the reasons is because microbes are present everywhere. So what I'm going to say here actually can apply to any ecosystem on Earth. Let's see now if it moves this. Can you see the next one? It's low?
Yes.
Okay. So basically what I'm going to say a few words about why do we care? Why do we study marine microbes in the ocean? Also, what happened in the last decades about the molecular revolution that changed the way we study the ecosystems and also the way we understand our planet. I will mention a few core molecular methods with the idea that most of you or some of you are not molecular biologists or microbial ecologists. So I will mention them and I will explain how you can use them, but I'm not going to get into the details. For that, you will have to come to a course that we teach every 2 years in Chile. It's called Ecology and Diversity of Marine Microbes. You are all welcome. Then I will dig into how you go from genes to function and activity. especially for this course, I will say a few words about how you can connect molecular data with satellite oceanography or satellite observation. And finally, I'm going to give a couple of examples just briefly and talk about future perspective, where the field is moving. The first thing you have to recognize is that the world, our planet, is full of microbes. From the surface of the ocean all the way into the ocean trenches where I'm working right now, and even sediments, everywhere you find microbes. But the problem was that until a few decades ago, we didn't know that there were so many in our planet, and I will briefly tell you why is the reason. So it was not until 1998, in a paper published in PNAS, that we got the first more accurate numbers of how many microbes there are in the ocean and in our planet. And it turned out that if you just consider the water columns, the prokaryotes, that means the bacteria and the archaea, they are about 10 to the 29th cells in the ocean. This is a huge, huge number. In fact, in one of my readings, I read that there are more microbes in the ocean than stars that we see in the universe. I told a colleague of mine, an astronomer, and in fact he said, "Yeah, that's right. Your number is bigger than ours." So that's a very, very big number. But and as I said, until a few decades ago, we didn't know that we have so many microbes in our planet, and that was due to a methodological problem, and I'll tell you about that. So when I talk about microbes today, I'm just going to refer to bacteria, archaea, which is another type of prokaryotic microbes, and then microbial eukaryotes. I will leave out the viruses, but just so you know, there are an order of magnitude more viruses than bacteria and archaea. That means that the number is 10 to the 30. So let's continue now. So again, microbes are everywhere. They are important. In fact, if you take the biomass of the living organisms in the ocean and you put them in a weight, they will constitute more than 80%. Before, we thought that we had a lot of fish and crustaceans and other organisms, but in fact, the microbes make most of the biomass in the ocean. And that means they make a big impact on things that are happening in this environment. So on one hand, and this is one of the things that you will see in this webinar series, is that phytoplankton, that means photosynthetic microbes that live in the ocean, they are the ones that are responsible for most of the marine primary production. A few percent are due to macroalgae, but most of it is due to phytoplankton. They fix carbon, they generate oxygen, they also act, or they are involved in nutrient cycle. They drive in the nitrogen cycle, sulfur and phosphorus, and basically any other element, including trace elements like iron, cobalt, or other metals that we need as cofactors in our enzymes. Microbes are also important for the carbon cycle. They process, remineralize, and export organic matter from the surface all the way into the abyss, into the sediments. And also for us as human beings that eat marine products, They are the ones, the microbes are the ones that sustain trophic networks and regenerate nutrients. In summary, microbes underpin major ocean biochemical processes. But if you go anywhere in the ocean, these communities are not static. That means that if you go one day and you go the following day, you may not find the same community. These communities shift, their composition shift, especially with gradients. With depths, the microbes that you find at the surface are not the same that you find in the deep ocean. You change with latitude. We know now that the diversity and composition changes from the equator to poles. They change with seasons through the year. They change with water masses. They also change with the geochemical parameters like oxygen and nutrients. And also they change if you consider structures like fronts and or if you have in an upwelling system, you will find different microbial communities. So patterns in microbial diversity reflect environmental structure through space and time. Let me just try to move this. So when we want to study the microbial community, what do we want to understand? We want to know who is present, which type of microbe is present in, for example, in a parcel of water. How abundant are they? What can they do? What are they doing now at the present when I'm observing them? How do they can interact? And how will they respond to change, for example, to climate change? And to answer these questions, there are different molecular methods that can be used to answer these questions. No single method can answer all of them. So traditional approaches in microbial ecology, this is when I was a student, both as an undergraduate and a graduate student, the methods that were out there was, of course, microscopy, which is very important and still in much use because you can actually have direct observations of the microbes. It has a limitation, their taxonomic resolution. Then the method that was very popular about microbial ecologies was to culture microbes, to isolate them from the environment, take them to the lab, and then you can manipulate and you can study them. But the limitation was that it was biased, this method, towards those microorganisms that you can culture in the lab, which it turned out to be a very minor proportion of the whole community. You can also use, or you could also, and still today, use pigment analysis, for example, if you study phytoplankton groups. One of the limitations is that you have a coarse taxonomic signal. And of course, if you want to understand biogeochemical process, you also do rate measurements. For example, you measure primary production, photosynthesis, photosynthetic rates. This gives you a lot of insight about what the microbes are doing, but usually, if you're working with the whole community, they are not taxon specific. So these are still essential tools in our field, but they capture only part of the microbial world. So what was the problem? The problem was that microbial ecologists, the way they studied microbes was to take a sample from the ocean, from gut content, from any part of the environment, and plate them in an agar plate, wait for them to grow, and then count them under the microscope. But then in the 80s, there was a method that is still being used today, that you stain your cells and you can count them under the microscope. So, and it turned out that when scientists did that, they found that there were order of magnitude more microbes in the ocean than what we knew by the cultivation problem. And this is, some of the reasons are because we didn't know what the growth requirement were of the microbes we wanted to study or isolate it. We gave them a medium, but probably they were not the ones that many of the microbes will use. The laboratory conditions, for example, if you want to isolate microbes that are, adapted to depth, and you grow them at surface depths, then you were not giving them the appropriate environmental condition. So the culture collections, even today, that we have, really underrepresent what is out there in the field. The current estimate is that we have only culture between 0.1 and 1% of the microbes that are in the environment. That's a very, very low number. That means that there's a big diversity that is out there that we don't have them in the lab. We cannot study them, physiologically, to do physiological work. So that means that the view we had on the microbial world was very limited until the molecular revolution. So just a hint of what happened, and this is something that has happened only in the last decades. So pre-1970s, we were using microscopy, cultivation, and it was basically based on morphology. And until that time, actually, microbiologists or environmental microbiologists did not talk to zoologists or botanists because they could not use the evolutionary theory to study microbes. Because although we could describe it, we can have them in cultures, and we knew some of their metabolic properties, we could not establish evolutionary relationships. It was not until the late 70s when Carl Woese, he was a microbiologist at the University of Illinois, he was for 20 years asking the big question: what is the evolutionary relationship between microbes? And he developed a whole framework to look at molecules to actually establish the relationship, the evolutionary relationship between microbes. He was a giant in microbial. He completely changed. It's a fascinating story. Many of the big scientists of that time did not believe him at the beginning. And one of the big discoveries that he did, or one of the most important, is that he found that within the prokaryotic world, that until then we thought they were all were bacteria, he discovered there was a whole domain, new domain of microbes, which we now call them archaea. At that time, he named them archaeobacteria, which is a completely different organism in terms of physiology and evolutionary radiation. Then in the 80s, a big step happened when a biochemical technique, the PCR, was developed. And this technique, what it does is uses an enzyme that can amplify a particular gene and make many copies of it that you can later sequence. Carl Woese did not use PCR. He used a much more uh, difficult or time-consuming method. So PCR really was the technique that sparked the use of molecular techniques in science. Then in the 90s, there were some techniques also based on PCR and microscopy, combining them, that started giving us a picture about the diversity that was in the environment. The good thing was that with this technique, you could use genetic targets in the environment without having to culture them. So you can go to an environment, do PCRs, select a taxonomic marker. I will get into that in the next slide, and then see what was the diversity in that particular sample. Then in the 2000s, again, another revolution, which was the high throughput sequencing, which opened the community scale analysis. You could get many sequences. You can sequence very deep and start looking at the microbial community that exists in any environment. So with the application of these molecular techniques, that revealed that most of the marine microbial diversity and function had been invisible to culture-based approaches. And that really created a big revolution in our field. So I'm just going to mention the method. This is not a course in molecular biology or molecular ecology. And just explain a little bit a couple of them, which are the ones that are most in use today. I mentioned PCR, which amplify and quantify specific genes. I'm sorry, it's called PCR or quantitative PCR. So this is a technique to detect the presence of a target taxa, for example. We have a sequence of our favorite microorganism, and then we can see if that is present or not, and how many copies of that gene are in my sample. You can apply gene abundance, as I said, how many of them, and also you can use this. For example, if you're interested in nitrogen fixation, you can use a gene, NIF-H, for example, and you can ask to your sample if nitrogen fixers are present and who they are. Or if you want to know, for example, if there are ammonium oxidizing bacteria or ammonium oxidizing archaea, you can also have developed a protocol and ask if the genes are present. For that, of course, you have to understand the biochemistry. You have to know which genes are doing what, and then you can ask your environment. A very popular method right now is amplicon sequencing, also is called metabar coding. Here, what you do is you use a taxonomic marker, and I'll explain a little bit more in a couple of slides. For microbial ecology, we use the 16L ribosomal RNA gene for the prokaryote, archaea and bacteria, and the 18S for the eukaryotes. And also, if you want better resolution, if you want to distinguish between very close relatives, or you want to study fungus, for example, you use another taxonomic marker that is ITAs. With this technique, you can get many sequences relatively fast, and you can see a lot of articles coming out using this technique now. So you can easily look at the community composition, you can look at diversity patterns, you can use this technique to do biogeography, to see where each of the microbial groups are for example, in the ocean. There's another method that was very popular. It's not that popular today because it's very time consuming. You have to use microscope, but it's very good when you want to count a particular microorganism and you want to see it under the microscope. It's called fish fluorescent in situ hybridization. And what you do is you add the probe with a fluorescent probe that only target your specific organ that you're interested, and then you can look at it under the microscope. For example, if you're interested in looking at symbiotic relationship, you can develop probes to look at the host and the symbiont. You can look, for example, into a microbial mat and look at the spatial organization. And so actually if you want to look how microbes organize in the environment, for example, in a particle or in a fecal pellet or as I said, a symbiotic relationship, this is the technique that you will use. Then a few years ago, it was very popular to use DNA fingerprinting. Again, you will do PCR and use techniques that such as DGG or TRFLP. They are not so much in use these days, but this was before amplicon sequencing, but it was to do community comparison, and this was just before amplicon sequencing appeared and it was in use. So with these methods, you can answer who is there, how much of them, and where they are. Now, there are another molecular methods that go from potential function to activity. And you probably have heard these names. Metagenomics, in this case, you don't use PCR, but you actually sequence you take the DNA and sequence the entire community. So that means you get the gene content. And these genes will tell you the potential, for example, the metabolic potential of your community, which biochemical process they could perform. They don't tell you that they are doing it, but at least that they have the potential to do it. They carry the genes of doing certain processes. Then, if you want to see which of the genes are actually being expressed at a certain place or moment, then you use another technique that is called metaproteomics. So that means that the genes that are actively being expressed. One step forward, and that's where the community is moving right now, although it's still not it's not used broadly because it's a very expensive technique so far, is metaproteomic. So instead of looking at the genes, you look at the actual proteins that are being produced and used, like enzymes. And then there's another technique that it's also not cheap technique. It's called single cell genomic, which in this case, instead of looking at the genes of a community, you actually isolate by flow cytometry or individual cells, you amplify the genome, and then you get the partial genome of that cells. Then if you're looking into function, also, you can, for example, take your sample, incubate them with C13 or N15, and then you can define probes, and you can apply techniques to tell which of the cells are using these elements. For example, if you add them C13 bicarbonate and you incubate it in the dark, you can say who of the microbes are doing dark carbon fixation. So it's not an easy technique, but it's something that is used to connect the molecular world with the biogeochemistry. So in this case, these molecular methods, they tell us also who is there, but also they can tell you what they can do and what they are doing at the present time or a special space. Okay, just a brief introduction or the basic idea that Carl was, again, developed in the late 70s, early 80s. As I mentioned to you, until then, we couldn't establish evolutionary relationship between different microbes. And the reason was that we didn't have enough morphological characteristic to classify them and put them into the tree of life. So what Carl was thought and developed, what he thought, I said, okay, what can I use a molecule that is present in all the organisms in our planet, where I can establish evolutionary relationship very distant in the past, but also in the present. And he realized that, or what his thought was, okay, all living organisms have ribosomes. These are the factories where you produce your proteins. And this structure, the ribosome, has to be very stable, because if it doesn't work, then you wouldn't produce your proteins, your enzymes. So it has to be very stable in time, varying but slowly. And this ribosome, which again is in all organisms, bacteria, archaea, eukaryotes, it's composed of 2/3 of RNA, structural RNA, and 1/3 of protein. And one of them is, if you look on your left in the back, is a small unit which is composed of mainly for RNA, and this molecule is structural, and then if you sequence that, it has regions that are very, very conserved. The length of this gene for bacteria and archaea is about 1,600 nucleotides. For eukaryotes, it's 1,800. So that gives you enough statistical information and to ask these molecules and to compare them among organisms. So the interesting part and the good part is that this molecule, this gene, has regions that are very conserved. That means that they vary very, very little in evolutionary time. And also you have regions that are very variable. That means that they are mutating, they are varying very fast, which allows you to establish or to use it to compare organisms that are more closely related. So this actually RNA gene sequencing gave microbial ecology an evolutionary framework. Before this, again, we couldn't put microbes into the tree of life into the way we were classifying or understanding the taxonomy and the evolution of animals or plants. So they didn't talk to each other. So the sequencers are identifiers of organisms, but also, and the important thing for environmental microbiology is that you didn't need to culture. You could go to an environment and you can isolate this gene and then use it to see who is present or not. So basically, how is it done? So the question we want to know is how the organisms are evolutionarily related, and are they new, or do they represent new taxa? So what you do is you sequence a molecular marker, for example, using PCR and then sequencing, such as the 16S, that this ribosomal RNA in bacteria and archaea, or the 18S for the eukaryotes. Then you align your sequences, and I put an example there. So you have organism 1 and organism 2, and you align them and you compare, each site of the nucleotide. And in that example, for example, you see that on the third position you have a G in organism 2 and A in the organism 1. And then later you have a T, and then you don't have anything. And then with this, you estimate the evolutionary distances. If you add the same species, they will be identical, these sequences. The more different they are, the more changes there are, the more distance they are in evolutionary framework. And this is an objective way of measuring evolutionary distance. So you go away from morphology, which is good when you can have characters that are inherited. But in this case, you use gene to tell you about these evolutionary distances and evolutionary tree. Then you can construct a phylogenetic tree which best represents this relationship, and there are a lot of algorithms and that are used again in a more objective way, and you can apply statistics to tell you whether your tree is how good it is in terms of statistics. And finally, you can also use it to identify potential new taxa. This is the way that new organs have been discovered lately, if you cannot isolate them. So if you do this exercise and you find that there's a very distinct clade or sequences, then you can propose that you have a new taxonomic group. Of course, until you, again, get it in culture and describe it, it will probably won't have a name, but at least you know that it is there in the field. And then you can use this to target and use other technique. For example, once you know this, you can develop a probe and use, for example, single cell genomic to get at the genome without having to culture them. The other way is that you can use this molecular taxonomy, and this is not only used for microbes, now it's being used for all organisms, for fishes, for mollusks, for everything. Is again, you sequence your molecular marker, and then you compare the sequence with a reference database to identify the closest known taxonomic group. For example, here I put an unknown sequence that I have that I don't know who it belongs to, and then I compare, and it turned out that it's closest to Prochlorococcus and Synechococcus. But if you see from that example that the sequence, my unknown organism is closest to Prochlorococcus, so I will infer then that what I have, that sequence, belongs to Prochlorococcus, the cyanobacteria, and is close to Synechococcus, but in this case, is closest to Prochlorococcus. So that means that now I can sequence many of them and assign a taxonomic identity to my sequences in the field. You can also, sometimes you cannot get to species because you don't have, they are not in a database. One of the caveats here is that your taxonomic assignation is very dependent on the database that you have. Because if your organism is not in the database, then you won't find a match, and you will probably say, "Oh, this belongs to a higher taxonomic hierarchy, like a family and order." You cannot get all the way to species. So one of the good things in our field is that we have, every time we publish new sequences, we have to deposit them in a data bank. Otherwise, your work won't get published. And that is very good, because that can tell you, if your sequence or the organisms that you have has already been seen in other places or is a new one. So again, what make all this possible, apart from the develop of the framework, the theoretical framework, and using Arboson, it was this PCR is now a common instrument that you have in almost any microbial lab in the world. And what it does is just used to get environmental DNA. You amplify the genes through biochemical technique with an enzyme. So you make many copies of this gene, and then you can sequence them. So we have gone from limited sequencing when we started working in the field to really massive data now. With the new technique we have throughput sequencing, we have thousands and millions of reads for each sample. And the field has moved very fast. So now with the new technique, actually, what we can do is we can start doing gene inventories or microbial inventories at the large, using to identify large-scale patterns. Again, one of the techniques that is most used today by environmental ecologists is amplicon sequencing. You take your sample, you extract your DNA, you PCR your taxonomic marker, 16S for bacteria, 18S for eukaryotes, and ITS also is used for some eukaryotic groups and fungi. You sequence massively, and then you start looking at using statistical analysis. You look at diversity indices, and you start doing comparisons. You also can use what is called metagenomic. In this case, you don't do PCR, you extract the whole DNA, you apply a technique where you shotgun, you cut your fragments DNA, you sequence, and here, since you have the pool of genes, you can do metabolic reconstruction, or you can even construct some genomes from your community DNA. So this is based to look at what is the potential, what the genes are in the environment that could perform a certain function. So that means is the potential. So what has been our conceptual transformation? So from culture organisms that we could isolate it, we went to studying whole communities. We went from just knowing their morphologies to their genetic lineages, again, put it into evolutionary framework. We went from just present and absent to actually the functional potential. And then we went to inventories, static inventories, to dynamic activity. So we're starting to look at the structure and factor of entire communities. So now, if you are in the field, what you do, you go and sample. If you're interested in phytoplankton or bacteria or archaea, you filter, you preserve them, you extract your DNA, then you make your library preparation, your sequence, then you do your bioinformatic analysis, and then you do your ecological interpretation. So now we're moving to integration. See, I mentioned that there are different uh, methods looking at the whole community. So you have metagenomic, who is there and what is their potential, metatranscriptomic, what that they are expressing, metaproteomic, what they are producing, enzymes, proteins. Metabolomics also is another technique which you can identify the molecules that, for example, are being exchanged between one organism and the other one, and also you integrate that with your rate measurement. And then, so in order to have an ecological interpretation and integrate it, you need to apply different technique. Now, what it has to be clear is that if you have just DNA sequences, you won't get very far. In order to interpret them, it's essential to have the environmental context. You need to know where your sample comes from in terms of, nutrients, temperature, oxygen. Otherwise, you won't get a clear understanding of what's happening. You can say who's there, but you cannot go further than that. So it's very, very important that you consider your metadata when you take samples, your molecular samples. So what molecular approaches what can observe? So as I said, I mentioned some of the methods. So you can, for each sample, you can look at the microbial identity, microbial community composition, what type of ecotype, genes, metabolic pathway, expression, and activity. So now I want to, what satellite observes? This is something you are learning from your other website, so I won't mention them. So now, where the field is moving is combining approaches. You use satellite oceanography for synoptic view, repeated observation, all things that you are learning in this webinar. And with the molecular approaches, you can identify the taxa that are there, reveal their genes, link communities to biochemical process. So this will give you going from to link, go from different scale. You would start dealing with mechanisms, also prediction, and then we're going to adaptive sampling. So the main gain is scaling mechanisms from sample to actually ocean basins. I'm going to give two examples. Heather is here. So actually, one of the things that I reflect about this is that many, many years ago, when Heather was a postdoc in my lab, actually, she applied molecular technique, PCR-based technique, to start looking at the distribution of cyanobacteria, the Prochlorococcus, in basic scale environment context. So the question was, were genetically distinct ecotypes associated with large-scale environment gradients? The approach was to use PCR-based ecotype detection, and the finding was that different ecotypes occupy different environmental niches, and this was basically controlled by the hydrography, mixing in the surface layer, and light. It turned out that it was one of the first works that connect between molecularly defined population with basic scale environmental structure. So it was a very early proof that molecular diversity can be linked to ocean scale structure. The other more recent example, now this one is based on this more used technique now, which is metabar coding. So what they did is that they use samples that had been sequenced, the entire community of eukaryotic plankton. They use metabar coding using the 16S of eukaryote. Then they use satellite observation and statistical and machine learning models to be able, if they can predict molecular now defined plankton communities at the global scale, combining, again, molecular data, now massive metabar coding data, with satellite data using these machine learning techniques. They were able to identify 6 eukaryotic plankton community that they could relate to the environmental context and reproduce them during the year. So again, this is where we start seeing the combination of molecular techniques with now satellite oceanography and models. Just finishing, 2 more slides. So where is the field going? So I already mentioned that you don't only apply one technique, but you're actually integrating omics. You're looking at genes, you're looking at transcripts of RNA, proteins, metabolites, and then you start combining them with rate measurement to start looking and integrated the knowledge about each of the communities. Also, we all know that if you go to the ship, it's very slow and very scarce now. A lot of they're applying to autonomous and in situ sampling. That, for example, they are putting instruments there that can filter and collect water through a front, for example, in autonomous, without having to go with a ship. So you send your glider, for example, and collect the DNA or RNA, you fix them, and then you return it to your lab and analyze it. Then you can also use satellite to guide your sampling. If you're studying, for example, an eddy or studying or a front, you then combine that with satellite information. There's a lot of new techniques now, machine learning, but also artificial intelligence is starting to be used in terms of predicting ecology, linking microbial to taxonomy or to biochemical fluxes, and basically, and also finally, what we want to do is do prediction. You integrate into models, and then you can say, oh, what's going to happen, for example, in 20 or 50 more years with climate change, and how that could impact the microbes in the ocean. So this is again what we would like to see. I'm finishing one more. Is an integrated observational framework. You use satellite to detect the feature that you are interested in. You use ships or autonomous vehicles. You collect your samples, you sequence, you analyze them, and finally you integrate this into models that allows you to make predictions. So this is last one. So what are the take-home messages from this lecture? The marine microbes drive major ocean biochemical cycles, that molecular methods have transformed our view of microbial diversity in our planet. Different methods reveal different layers, identity, abundance, function, and activity. Combining molecular observation with satellite oceanography allows you to scaling from samples to the ocean, and the field is moving toward integrated predicted microbial oceanography. Thank you. Gracias.